Good evening, everybody, from a very stormy Kittery, Maine, and I welcome to Shoals Marine Lab's weekly marine seminar series. I'm Jennifer Seavey. I'm the executive director of Shoals Marine Lab. For those of you who are not familiar with us, we are the oldest and the largest undergraduate focused marine lab located on Appledore Island, about 6 miles offshore Portsmouth, New Hampshire. Every summer, we host this seminar that we affectionately call the Rock Talks for our island community. And this year, in light of the public health emergency that we're in, we have put our courses and this seminar online. So we have taken it off the island and brought it to you. And we're so glad that you could join us here tonight. So our format this evening is a 45-minute talk, more or less, followed by 15 minutes of Q&A. If you want to ask a question, which we would love for you to do, please use the Q&A box, which is at the bottom of your screen. You can type in a question, and then I will help facilitate that with Jarrett when we get to that part. If you have any technical needs, also put that in the Q&A and we will help you there. Just one more note, since my power keeps flickering on and off, we're in 3 different locations, the 3 of us sort of leading this call. So I think we'll be able to pull it off no matter if one of us, well, obviously if Jarrett shuts down, then we'll all be shut down. But Colin and I are sort of replicating each other tonight. So just wanted to warn you that that could happen, and that might happen for some of you too, because a lot of you are calling in from the local areas. So we'll just be flexible with what happens. So I'm really excited tonight. Dr. Jarrett Byrnes is here with us. He's an assistant professor from UMass Boston. He is a Shoals alum, faculty member, researcher. He does it all. He's all Shoalsy, and he does some really cool work that you're going to hear about tonight. So he did his undergrad at Brown in biology. He has a PhD from UC Davis in population biology, and he did a couple of postdocs, one at Santa Barbara's long-term ecological research station, and also at the National Center for Ecological Analysis and Synthesis. Jarrett focuses on the causes and the consequences of complexity in nature. He is interested in how humans alter diversity and the interconnectedness of life on Earth. And he explores these topics in salt marshes, which he's been doing a lot of this summer, and also kelp forests, which he's going to be talking to us tonight about. So tonight we're going to learn about kelp forest ecology around the world and then bring it right here to Appledore. So without further ado.
Hi, everybody. Let me share my screen and we can get this shebang on the road. So first off, really wanted to thank Jen and everybody for inviting me to give this talk. It is a true delight and honor to give a rock talk for the Shoals Marine Lab. So I'm really happy to come and talk to you today about some of my work in kelp forests. I've really tried to cater this towards the work that I've actually been doing out at Shoals. So for those of you who have seen me out and around there over the past few years, or might in the future, you'll actually know what I'm doing. But you know, again, I just want to say what an honor it is and how delighted I am. I've got a long record at Shoals. I've been coming out there since the late 90s. I was a student in 99. I took experimental marine ecology with Doug and Denny and Sarah and really learned to love ANOVA. Got some great experience underwater with Jim, and that really was the class that kicked off my whole career. And then got to hang out and learn quite a bit about invertebrates with Drew Harvell and her invert zoo class. So it's, you know, it's a long history. From there, I got to spend some time at Shoals the following summer as an REU, studying convoluta. And this is actually an old picture of all of us. It's the only picture I could find of that first cohort hanging out. And I kid you not, I'm lecturing about 2A ANOVA. So the more things change, the more they stay the same. And then Jim, for whatever foolish reason, invited me to come back and TA the following summer. I spent some time after that in the wilderness, so to speak, trying to figure things out. And obviously, you know, it didn't work out too well. And was delighted at the end of all of this to come back and be able to get into the water, and have been doing research on kelp forest out at shoals since about 2013, and hope to just continue that on. I'm also, again, always honored to be part of the shoals family. I've been privileged to step into the enormous fins of Jim Coyer and teach underwater research out at shoals. And so some of you who are on, who are students, who are thinking of coming out next year after you get your C card and learning underwater research, hopefully we'll get to run into each other. Okay, so that's the background and the mushy stuff. Let's hop into some of the science. So today I'm going to be talking to you about kelps. For those of you who are unfamiliar with kelps, kelps are big, bold, beautiful brown algae. There are a wide variety of different types of kelps. You have canopy forming kelps, you have kelps that are rather short with long stipes, you have prostrate kelps. I know that many of you, when you may think of kelps, immediately you begin to think of the great cathedral-like giant kelp forests of California or Tasmania or New Zealand. That's a sort of the common locked in our head conception of what kelps are like. Around Appledore and the Isles of Shoals and here in the Gulf of Maine, kelps are quite different. Instead of those giant redwood forest-like wonderful ecosystems, we instead have these lush rolling meadows of kelp that are, again, just a very different ecosystem. They're beautiful. They're like unto the prairie, really, most of all. So it's really just a wonderful, wonderful system to work in, very different. We do get some pretty big kelps, so they can be quite large, not quite giant kelp size, but we do get some rather big kelps here as well, and they are an incredibly important species within the system. Kelps are important across really the entire planet. They cover roughly 25% of the world's temperate coastlines. We often hear the phrase, you know, why is the world green? Well, why is the seashore brown? It really is brown in temperate ecosystems throughout the world. There are a wide variety of different genera, a wide variety of different species that make up kelps, as it were. And yet wherever they are, these large structure-forming brown algae have a huge impact. Those of us who have taken an ecology class or even in Biology 101 or even in high school biology quite often may be familiar with kelps from the classic paradigmatic Aleutian otter, Urchin, kelp, trophic Cascade story, where you had in Alaska and throughout the West Coast, otters were hunted out due to the Russian fur trade, driving an explosion in urchins and then leading to the denuding of kelp forests. Over time, as otters recovered, that reversed itself. And I know those of you who are at Doug Rasher's Rock Talk have also heard a great deal about what is going on within that system as well. So I'm hoping that you're ready and willing for another system of kelp to come your way. That urchin phenomenon is actually quite common throughout the world. We are no stranger to it here in the Gulf of Maine and really the whole Western Atlantic. Urchins can create an enormous impact on kelp forests. In fact, in 99, 2000, 2001, while I was out here, a great deal of Appledore was covered in urchin barrens, and I'll talk a bit more about that later. It's really been quite shocking to me to come back and work at Appledore again and find that that is no longer the case. Kelps are a paradigmatic foundation species. They provide habitat for a wide variety of fish, invertebrates, commercial species, non-commercial, etc., by providing a great deal of structure. They provide structure not only in the enormous individual blades and stipes that go up to the surface, but kelps root down to the bottom, or root, if you will, by holding onto the rock with a holdfast, and within that holdfast itself, you can find a tremendous diversity of life. Juvenile organisms, smaller microcrustaceans, and a wide variety of other species. So again, like you think kelp, you think habitat creation, huge ecosystem service that they provide. Kelps are also sort of a paradigmatic bottom-up control species. And what I mean by bottom-up control is that kelps really feed the whole food web from the bottom up. Nutrients come into the kelp and then feed the rest of the system. And they do that through a wide variety of different ways. Of course, some species do directly graze on kelp, such as this kelp crab pictured here. But you also have a wide variety of species that will feed on kelp detritively. And that's because in many systems throughout the world, kelp grows so fast that it is continually shedding off biomass, such that species that would normally be active grazers, such as these purple urchins, end up just sitting back fat and happy and eating the detrital rain that falls upon them. Kelps feed a lot of fish food. You have a wide variety of different micrograzers, amphipods, isopods, tiny snails, etc., that will consume kelp and live within it, and that feeds a lot of the commercially fished species or non-commercial fish as well that we see out there. And in kelps also slough off a huge amount of mucus, feeding the whole microbial food web around them. In fact, if you go in and look at fish within a kelp forest, so here we've got this wonderful work from the Santa Barbara Coastal LTER, and you look at the mean kelp carbon contribution across the x-axis and the mean trophic position of fish on the y, you can see as you climb up and up and up the food web, as it were, you're seeing that kelp-derived carbon magnifying and amplifying, and you're really seeing that kelp sits at the base, but it ultimately feeds the top. Not only that, not only does it feed things local to itself, the impact of kelp forest is far and wide beyond the forest itself. So I mentioned before that kelp shed off an enormous amount of their biomass. Kelp has been found in the depths of the Monterey Canyon, feeding detrital food webs. You find it offshore of Nova Scotia. You find it in a wide variety of different deep water habitats, where if you look at this figure from Krumm-Hansel and Scheibling, you can see that detrital production and productivity rivals any other ecosystem in the ocean. It just really powers these temperate ecosystems in a deep and fundamental way. Not only is it important in temperate ecosystems, but kelps also, of course, have their use in human systems. There are, of course, many long traditions of eating kelp. This is actually a picture that I took at a kelp harvester's home in Japan in 2015, as she was grading some of these kelps before sending them to market. So you have a very, very long tradition of both kelp harvesting and more recently kelp farming. You can, of course, pick kelp up as kombu in your local market. This is typically a laminaria or saccharina species. And often you'll even find kelp sliced up thin and re-dyed green and put into your seaweed salad. So those of you that have had seaweed salad, a lot of that's agar, but quite often you'll also find some kelp tossed in there as well. In fact, here in the Gulf of Maine and throughout New England, we're witnessing a burgeoning seaweed industry. We have kelp farming really taking off. We have a lot of events that have been happening over the past few years. This is from something I went to in 2014, where I got to have some kelp dusted bagels that were delicious, as well as some various kelpy ice cream flavors. It's really an industry that's taking off, and there's a wide degree of investment in trying to see how can kelp end up becoming part of the working waterfront throughout New England. Now, this is all well and good. I'm like, kelp, it's the super alga, and it is. But we also know that there are threats, at least to natural kelp populations. So this is a figure published a number of years ago by Ben Halpern and colleagues, showing cumulative human impacts within our coastal oceans. So red colors are very high impacted areas. Cooler colors are lower impacted areas. And if you look at the distribution of impacts where kelps are present, so rocky reefs, you can see that kelps tend to exist in a lot of that space that has a medium to very high impact. There are some kelp beds in areas with lower impact, but overall, that average of 4.75 is pushing it into impact, and a wide variety of reefs exist in the tail of that distribution and are under threat. So today I'm going to try and tell you a couple of stories about kelp. I'm going to focus on first, how is global kelp abundance changing? Do we actually see changes across the planet that is consistent with accelerating human impacts? And then I'm really going to focus down, particularly for you all who are interested in the Gulf of Maine and ecosystems there. I'm going to talk a bit about what is potentially driving variation in the Gulf of Maine from some work that I've been doing with some collaborators. And then I'm going to do a deeper dive into, is temperature really one of the big drivers here within the Gulf of Maine? And what does that mean, not just for kelp? So I'm going to stay very alga focused for a while, but I want to get into what might that mean for the rest of the community, given that kelp may be a real foundation species here. And then I'm just going to have a little coda at the end. This is something I inserted last night because I think it would be really interesting to start to think a little bit more deeply about what's going on at the small scale of Appledore itself. And this is some historical work that, again, Jim Coyer exposed me to, and I was lucky enough to go around and do some surveys with him as well. It's stuff that's not worked up, but I think it's stuff that tells a really interesting story about this once and future Appledore. Okay. So let's start with, how is global kelp abundance changing? Let's start with a global story to create context. So the sort of framing question here is, have we observed changes in kelp abundance? Have we observed areas across the planet that have gone from, say, a kelp forest pictured at the top to sort of a foliose algal bed pictured at the bottom? Have we seen this? There are a wide variety of reasons to predict why this may have happened and that it may be common. And a number of years ago, I was fortunate enough to lead a kelp and climate change working group at the National Center for Ecological Analysis and Synthesis, or NCEAS. And so at this NCEAS working group, we got together and said, all right, actually it was a fascinating process. We all came into the room fired up to think about, how is climate change altering kelp across the planet? And what we realized very quickly is, in order to ask, how is climate change affecting kelps, you first need to know, are kelps even changing in a consistent way across the world? And so our big first task was to set about and do just that. And we began by going through our own file drawers, going through our Rolodex of other kelp forest researchers, digging through the literature, both white and gray, and creating a global kelp database that we call Kelp Time. It's roughly 8,000 measurements of divers in the water surveying kelp. So these are all diver measured kelps. And the idea with this analysis was to take these field measurements of laminarian kelp, and we did specify laminarian, and can get into that in the Q&A. We tried to find a way to take the sort of panoply of different types of measurements that were out there, look at measurements, say, at different sites through time, and after standardizing them, ask, do we see consistent trends over time? And of course, we began with this a priori expectation that, yeah, you have climate change, you have a wide variety of other forces that are impacting kelp populations. I bet if we go through and use very high quality fancy pants statistics, so Bayesian hierarchical mixed models, to try and tease this apart, we are going to see that there is a consistent trend in kelp across the planet. That really was our expectation going into this. We found that there are a wide variety of different great sources of data out there, not only colleagues that we were able to work with or literature we were able to pull, but there are really some wonderful monitoring sites out there for things like power plants, for example, off of Plymouth, et cetera, or off of Seabrook. So here, for example, is some some data from Normando and Associates looking at the Seabrook power plant, and you can see percent cover of kelps over time, both at the discharge site and control site. And so you can actually see that trajectory. So we can really get some wonderful data from these types of different efforts. So we took a wide variety of data sets like this, and in the end, this is what our coverage of kelp around the world looked like. So this is a map of just where did we get the data from. Now you can see first off, a lot of this map is gray, implying that there's a lot of the world that we don't actually have good kelp data for. And I'd say that's a huge problem, and something that in the Q&A I can talk about some of the ways that we're trying to rectify. You can also see that where we do have data, there's some areas with cool colors where we have relatively few measurements, but we have some. And we have a number of areas that are real hotbeds of kelp monitoring. So off of California, in some areas of the Gulf of Maine, off of the UK, and in the Aleutian Islands, there are a lot of data sets there. So we realized this was a problem and we couldn't necessarily look at a global average. And so as I go through this and tell you more about the analyses, the results really are different for different places, because we didn't want to approach this data set with the hubris of saying we could arrive at a global answer when things might be far more regional and might be limited by different data coverage in different areas. Most of our data sets were from a 10 to 20 year trajectories, so they weren't as long as we would like. We would, of course, all love the 62-year data sets. That is a particular data set from off of Scotland. But we had a wide variety of different data sets of different length, but 10 to 20 years, that's actually a fairly decent time series to do some work with. Now, the problem we did sort of acknowledge with this data is that many of our samples were recent. So while they may be 20 to 30 years, many of these data sets started in the 80s, 90s, some even in the early 2000s. So this is much more a snapshot of change recently than over a very, very long period of time. That said, even over the last few decades, particularly in fact over the last few decades, we'd expect some really dramatic trajectories. So okay, keep talking about dramatic trajectories. I keep talking about the possibility of things being the same. Those of you that are used to a little setup and anticipation probably know where this is going. And that is that when we actually look at the data, we see a lot of variation. You know, I have hints of a decline. This is actually an old slide. I'm not even so sure of that anymore. You can see that each of these lines, and every one of these lines is a fit from an individual time series. The time series themselves are sort of ghosted a little bit in the back. And you can see that sort of band at the bottom, which is our overall average fit, which does show sort of a slight decline. But there is just a lot of variability here. It's all over the map. If you look at the slopes of those individual trajectories, so here I'm showing you the proportional change in kelp per year across our x-axis, and different sites are arrayed along the y. You can see not only the estimated slope, there's a black dot for each individual site, but those bars are credible intervals, 95% credible intervals. While on average across our data set, yes, there was a decline on average. However, again, you can see there's just an enormous amount of variability here. And we really wanted to be careful about that and make sure that our analysis was not dominated by a few regions. So if we take a step back and revisit the global picture, so we go back to sort of a spatial view or a map of the world to look at variability, shown here, I've got a map of the world. Anywhere with a warm or red color is somewhere where the estimated slope of the relationship for that ecoregion was negative, in other words, decline. Anywhere where it's a cool color, that's an increase. And I have some Bayesian probability cutoffs, just so you know that some of these regions you can have a high degree of confidence in. Others, your degree of confidence may be a little bit less. That said, as you look at this map, if you see no clear pattern and instead a lot of individual stories, you win the prize, because that's exactly what is going on here. There's a huge amount of variability in trends. Roughly a third of the world is in decline, a third of the world is, seems to be kind of hovering in stasis, we can't say one way or another, and a third of the world is actually witnessing increases. That's fairly striking. I will say for anywhere that there is an increase or a decrease, kelps are, you often hear the phrase canary in the coal mine. kelps are not the canary in the coal mine. They are nature's punching bag. They are the last man standing. They are the one that if it falls, you know something dramatic is happening. And so anywhere where you do see a fairly dramatic increase or decrease, that really says something. That says that something strong is happening within that system. And so while it seems at first like a wash, the fact that we do have evidence of strong change in so many regions is really very striking. But the variability is huge. So here, for example, we have our New England, Nova Scotia, and a little bit further north. So we've got a couple of different regions shown adjacent to each other. And you can see that there's loss and gain almost next to each other. In fact, in some ways, it almost looks like a gradient. And I'll get to that in a minute. But the big take home here that I'd like you to take from this portion of the talk is that if we ask, how is kelp changing around the world? One, it's variable. It's highly variable. We have this roughly a third, increase, decrease, no change. So there's a huge amount of variability, but there is change. And that there is change at all is really quite amazing. Again, kelps recover from disturbances like nobody's business. And that we can see either decreases or in some cases increases means that there is something dramatic changing those systems. So what is the driver of all of this variability? Where can we go with that? Well, for the rest of this talk, I'd really like to use the Gulf of Maine as a point of reference to begin to understand that variability. So let's talk about what is driving variation in the Gulf of Maine. So I showed you this figure previously. Why this variation? Throughout the Gulf of Maine and the Maritimes. So let's talk about this a little bit. I'd like to kind of just briefly go through the story of kelps within the Gulf of Maine in particular, but really New England writ large, as relayed by Bob Stenick in his sort of wonderful comprehensive history of kelp forests within the Gulf of Maine from 2013. And there the story is that previous to the 1970s, the Gulf of Maine was a rather kelpy place, full of Saccharina latissima, Laminaria digitata, Laria esculenta, and other species. But we were predominantly in a kelp-dominated system. One of the reasons for this was a wide variety of different fish species, and here I'm showing some wolffish, but predominantly cod, really ate a wide variety of invertebrates within the system, and in particular, the cod-urchin interaction was quite key to maintaining the system. In the early 1980s, or in the 1980s to the early 1990s, the system really flipped into a barren state. So you had urchin barrens throughout the Gulf of Maine. You had urchins, just like in the Aleutians, over-consuming a wide variety of kelps and other algae in the system, and you had pavements everywhere. And I'll tell you, when I was an RU at Shoals out in Smith's Cove, like the great thing about that site was that we could see those flatworms very easily against the pavement of these urchin barrens. And so it made it an awesome place to set up experiments, except for all of the urchin spines you would get in your knee while you knelt down on the rock. In the late 90s, though, there was a trifecta. One, disease. Throughout the Western North Atlantic, we saw a variety of different diseases, at least in some places that's been characterized and others it hasn't, that ended up impacting these high densities of urchins. Much like the diadema outbreaks in the Caribbean, when you get high densities of urchins, you tend to get disease. We see it here, we've seen it in California, we've seen it in other places as well. Second, you had the rise of the crustaceans. So cod weren't only eating urchins, they were also controlling a wide variety of crustaceans, and as their abundances went up, we also saw concomitant impacts on urchins. And third, there was the development of a fishery for our green urchins, which of course humans are very good at extracting, and that was yet a third thing that impacted urchin populations. And so you really had a trifecta of forces working against sea urchins and therefore allowing for the flourishing of algae that was formerly suppressed. And so you can really think of the Gulf of Maine as going through these regime shifts. You had the pre-1970s with sort of a more of a cod, urchin, kelp state. In the 70s to 90s, with the diminishment of cod, you had an explosion of urchins. And post-urchin fishing, as well as the other impacts I mentioned, you had the rise of really the crabs. Now I realize that's a blue crab there. I really need to change the slide. We're about to get the rise of the blue crabs. Been hanging out with them in Plum Island this week, surprisingly. Really, it was cancer crabs. So Cancer borealis, Cancer irroratus are really driving that. Ooh, gotta change that. That is embarrassing. Okay, so this sounds compelling. Why then, when we look at this figure, do we see declines and increases? Why isn't it all increases or all at least stasis? And I will tell you also, while he is a co-author on the paper, Bob Stanek doesn't like this figure very much either. In fact, if you look at his data in particular, you see within the mid-coast and further down east in the Gulf of Maine, you do see a lot of increases in kelps and not decreases. So there may be even finer grained regional variability than is present within this map itself. To get at that, Doug Rasher's postdoc, Theo Suskowitz, worked with Walter Addy and Bob Stanek, Doug Rasher, and myself to revisit a number of sites throughout the coast and look at change over time from York all the way down east into Maine. So the idea here was, if you can look at surveys done in 2004, as well as surveys that revisiting those sites in 2018, can we actually pick up a change in kelp, and do we see a consistent change in kelp over time, as I've just sort of shown in that map? Now, there's some interesting stuff about going along this gradient that Theo was very quick to notice, and that is that if you look at over time, something like degree days, or in other words, the number of days where the temperature is greater than 17 degrees Celsius, and this is true for a wide variety of different thresholds, you find that in the southern Gulf of Maine, York, Casco, et cetera, you start to have, you have a very clear increase. So you have very clear warming within the southern Gulf of Maine, whereas in the northern Gulf of Maine, while there may be some warming along the coast, it's muted, and in some cases, much more flat. And so you have a huge amount of variability in the temperature signal across the Gulf of Maine during all of this. So this seems really intriguing. And the prediction from this would be that you would have different trajectories of kelps depending on where you are within the Gulf of Maine and New England. That further south, you may be witnessing declines, but further north, you should see increases. And indeed, in comparing those surveys from 2004, from DMR to 2018, that's exactly what we see. We see that in the southern Gulf of Maine and warmer regions, decreases. So these are box plots of percent cover of kelp abundance over time. Right now, we're like really dialing in the models on these, so this is very preliminary, but I think we've got it nailed just about. Again, fancy statistics, it saves the day. And here you can see that there are some decreases in the southern Gulf of Maine and warmer areas. Then it flattens out, and in fact, in down east Gulf of Maine, we actually see increases. And so you really do see this rebound from urchin loss. You really do see this increase. And so we're getting contrasting patterns across the Gulf of Maine. Now, I've kind of been talking about this story of sort of a new trophic cascade, and I'm talking about temperature at the same time. I'm kind of talking out of both sides of my mouth. You know, the way this story seems to be going is that recovery happens, but potentially it's hindered by warming. Potentially you have this new state, and again, going to replace that embarrassing blue crab until it really does become the dominant delicious species within the Gulf of Maine. I'm very excited about that as a native Baltimorean, versus climate change that will hinder this recovery. But there's sort of another question. I just talked about, I think, a very compelling piece of data, but other things vary along this gradient. You know, the real problem in doing this kind of work is that while you can pick up some really interesting signals, we know that there are other things going on oceanographically in terms of human habitation and urbanization, in terms of a wide variety of other things in this gradient. And with 2 data points in time, it becomes very difficult to tease out all of those other drivers and start to understand, is this really a thermal signal that's altering recovery? Is there a temperature signal at all? So compelling, clearly something is going on along a spatial gradient in the Gulf of Maine. Probably temperature is at play, unclear, but most likely, yeah. Seems like a good story, and that's sort of playing havoc with the recovery of kelp in the face of the opportunity to flourish. So is it really temperature? And what does that mean for the whole community? I've been very kelp focused so far. My PhD is in population biology after all, but you know, let's think about the whole community. Let's step back a little bit. You know, is this really all temperature? Are there other consequences of change in kelp within the Gulf of Maine? And the reason I'm asking that, are there other consequences? You'd think that I would know off the top of my head, but here within the Gulf of Maine, while kelp is known to play some roles, there's not nearly as much research on the ecosystem functions and services provided by kelps as you find, for example, in giant kelp systems or other places where there's a much larger species of kelp that are more well understood. We really don't know our meadows nearly as well as we should. So to get at this, I've been privileged to work with what I call KEEN1, the Kelp Ecosystem Ecology Network of New England. KEEN1 is a group of researchers throughout New England that are part of a much larger framework, the Kelp Ecosystem Ecology Network, which grew out of that NCEAS working group as an effort to try and understand kelp forest ecology throughout the world, and really to understand the impact of global change on the world's kelp forest. So this is a wonderful network. And here in New England, we have our own sub-network. We meet annually, try and keep our methods standardized and try and do standardized monitoring to understand long-term change in the whole kelp forest system. So we've been doing surveys from 2014 through 2018, or rather, wow. Well, I'm going to talk up to 2018 here. We did surveys last year as well. This summer, it's a little bit more mixed given COVID. So it goes. And Keen of New England, or KEEN1, is really a consortium. Well, looking at this list, it's really a consortium of Rock Talk speakers. Myself, John Grabowski, Doug Rasher, Jen Dykstra, and Austin Humphreys. And we maintain sites all up and down from Rhode Island down to Damariscotta. So each of the black points on this is a different KEEN site or a collection of a couple of different KEEN sites. And what I'm going to be talking to you about today, and I'll talk a bit more about this in a moment, is the impact of temperature that we can discern by doing an explicit spatiotemporal design, that we sample the same sites, but we sample them multiple years in a row, not just at 2 points. So you can see the buoys and pulling data from there. Right now we're in the process of refining and trying to get slightly better temperature measurements a little bit closer to each of these sites. At each of our sites, so for example, at Appledore, we have 3 sites. And at every site, there are 4 stratified random transects that are sampled annually. So for example, here you can see we have a northeast Appledore site going from Broad Cove around to Devil's Dance Floor. We have a northwest site going from North Head all the way down to Sandpiper Beach. And then we've got a southwest site that goes all the way around to Smith's Cove. At each of those sites, there are 4 different transects. We've actually tried to match each of these strata within sites up to some of the long-term intertidal transects. And Kyla Byrnes, myself, and others are actually trying to find ways to sync this data up. So, you know, more on that in the next couple of years. At every site, we assess the abundance of over 215 different species. So we have fish transects, quadrat counts, swath counts, benthic uniform point counts, all along a 40-meter transect. We do it all. I mean, essentially, we we try and sample all the things. It's a very sort of exhaustive sample where we try and get as much information as possible about each and every site. So let's take a step back and begin to look at the data. So first, the buoys that we're using for temperature here show a great deal of variability in temperature in space and time. What's great about this is you can definitely pick up the spatial gradient in temperature. So the pink lines at the very top of this are indeed Rhode Island, and the gray lines at the very bottom, the cooler sites, are indeed up by Damariscotta. So there's a lot of variability in space, which is great. That's grist for the analytic mill. But there's also a lot of variability in time. There's a huge amount of temperature data there. Anyone who's doing observational work with time series of abiotic predictors knows it's a pain in the butt to take one of these series and try and downsample it and figure out the right element to look at. Within the Keen data, we've chosen to take a look at winter temperatures, and I'm going to talk today and why winter. It seems a little odd, but we're using winter temperatures because many of us are sampling at sort of the midsummer. And we know that kelps have this awesome biphasic life cycle where you have adult sporophytes, which is what we see in the oceans. Yeah, there we go. So we have adult sporophytes that put out zoospores, typically in the late summer and fall. Those zoospores generate gametophytes, and actually the real reproductive action and the seeding of new sporophytes happens in this area, which is the winter. And that's what's going to ultimately generate new individuals. That's also in the sort of later winter is when you start getting serious growth put on by a lot of these individuals. So this is winter, and that's what we're going to go with. I'm going to use winter minimum. It simply provides a better fit using information criteria, but there are a wide variety of different composite indices you could use here. And what you can see again is that there's clear spatial separation across the sites. But what's cool is that as you look at the winter minima that we're getting throughout the sites, there's a fairly consistent trend in increases in winter minimum temperatures. There aren't really any obvious trends in kelp. There's some more obvious trends in biomass, which I can talk about in Q&A, but the initial work I've done on this is largely focused on abundance and cover. The trends aren't as obvious, but once you start to dig into the biological data, and here's where I think things get interesting and why we need to think about the whole community, you can see here clear declines in total species richness. So this is the total number of species at these sites. Every different line is a different color. Again, we're looking over time. We have total species richness on the y-axis. And what you can see is that there are pretty clear declines in richness over time. What's going on? Is there a temperature link? Now, you could start simply by looking at the relationship between temperature and sugar kelp abundance. So I'm focusing on Saccharina latissima here. It's the dominant species at all of our sites. Some of the later analyses will use all kelps lumped together. We find a consistent signal whether or not we go with a dominant or what we see. And yeah, we see all of the species everywhere, but Saccharina latissima sugar kelp really is the primary thing we see across the Gulf of Maine. So okay, there's a negative relationship here, which is compelling, matches with Thew's work very well. But as I mentioned before, lots of things vary along this gradient. I mean, we've got a huge biogeographic break in Cape Cod, for example, as well as a wide variety of other different oceanographic phenomena. So huge amount of variability. What do we do? What's interesting, for those of you that are just starting out in the sciences and are starting to realize how many wonderful observational data sets are out there for you, I envy you. And that's because we're in the middle of a causal inference revolution in many ways. The techniques I'm going to talk about here are drawn from econometrics primarily. I'm going to talk briefly about two techniques, and I'm happy to, well, you probably don't want to get me started in talking about these techniques because I really will go on forever. But there are some really interesting ways out there that have been derived by fields that can't do experimentation to start to tease apart clean signals of drivers that you're interested in. And so the first technique I'm going to do is I'm going to separate apart the mean site temperature. In other words, that sort of spatial gradient. What's the average temperature in the site over time? I'm going to pull that out as its own variable. And that variable stands in as a proxy not just for temperature effect, but also anything else that varies along that gradient. And then I'm going to look at the site temperature anomaly as my actual predictor of a clean causal link with temperature. So if we do this, we can use a technique like structural equation modeling, which allows you to look at the sort of direct and indirect effects of different drivers throughout a system. So here we've got site mean temperature and temperature anomaly. I'm going to fit a model where we look at how both of those influence the cover and density of kelps, which provide different metrics of what kelp is doing in the system. And we know typically that as you increase the density of kelp, you also get an increase in cover. So I'm going to include a relationship there as well, so we can understand, again, these direct and indirect relationships. And then finally, species richness. I'm going to ask, does kelp influence species richness? Is this sort of a set of indirect effects where warming influences kelp, which influences species richness? We find that in California and other systems, where we find other abiotic drivers indirectly influencing species richness. Or is it direct? Does temperature somehow influence species richness? So in these models, we also incorporated percent cover of rock and a random site effect as well, for those of you mixed modelers following along at home. Okay, let's talk about the results. So let's step back again. We've got our site mean temperature, which is a proxy for maybe temperature, but also anything else that varies along that gradient. And so we can we can look at it and it's interesting, but it's not very meaningful, versus temperature anomaly, which has real causal meaning. So first, we find that site mean temperature affects kelp density. Warmer sites, and I mean warmer on average, tend to have more kelp, or sorry, tend to have less kelp. So Rhode Island has way less kelp than you have, out at shoals. However, we also find a temperature anomaly effect. So the clean causal estimated effect of temperature is there. In this analysis, we find about a 16% decrease in kelp density per degree increase in our winter minimum temperature. And yeah, we find that kelp and percent cover are positively related. But the kicker is that species richness appears to be driven by temperature, that as things warm up, richness decreases. It's not related to kelp. This seemed suspicious to me the first time I ran it. And so I looked at it through a number of different lenses, particularly to try and understand the implications of this causal framework I'm using. And I went back and ran something way more conservative that made me throw out a bunch of my data, but I could have even more trust in. And that's an analysis of change. Do you see species richness changing with change in temperature and change in kelp? So I ran a model that looks like this. So we have change in temperature, change in kelp, change in species richness. And again, is it a direct or indirect effect? And I expected to see at least some indirect effect. But if we build through again, change in temperature influences change in kelp. Again, this is a slightly different data set because of how it's filtered for this analysis, but we see about the same amount of loss in kelp per 1 degree C of warming, about the same goodness of fit. We don't see that indirect effect again. We don't see the kelp species richness link, but we do see a direct effect. So to recap, if we look at change in that winter minimum temperature and percent change in kelp, we do find a really nice, strong, consistent effect. Richness, however, and I've run this looking at richness of mobile invertebrates, sessile invertebrates, algae, total species richness, really have not trusted this result, and yet I can't really find a signal of kelp on richness. This depressed me for a while. Because this seems to fly in the face of a lot of the paradigms about how kelp forests work. And yet it's actually interesting. I found precedent for this, in particular as you go back and look at Shoals alum Phil Levin's work on Cunner. You find a wide variety of elements in Phil's work showing that it's not kelp per se, but total algal cover or total algal biomass that really matters in regulating at least cunner abundance and the abundance of some other fishes. That there's something slightly different here in the Gulf of Maine, that our kelps may be similar enough to some of the other species they compete against, and that can fill space when they come in, that the story in the Gulf of Maine is, dare I say, more complex than we find in other kelp forest ecosystems. This gives me enormous satisfaction. You always like to know that your system is its own unique and special place, and I think it is. I think we have much to learn about what exactly is a foundation species from the kelps here in the Gulf of Maine. But to sum this portion of the talk up, warming will reduce kelps over time within the Gulf of Maine. We're seeing evidence of that already, and we can actually find a clear causal link between temperature and kelps. That warming will likely also reduce species richness. There's something else going on here with respect to species richness, and I think there is a lot of room for research to try and understand what's going on here. And lastly, and I think most intriguingly, that kelp may be a little bit different here within the Gulf of Maine than it is in other places of the world. I, for one, am really curious to figure out how and why, and I think that's something of a priority going forward, particularly if we're going to manage our system properly. To what extent, if by 2080 we are kelp-free within New England, as is predicted by many of the models, how are things really going to change? Are they going to change in a meaningful way, or is there some redundancy or other elements built into the system that we should be aiming to understand? All right. I'm going to end on a slightly different note. I think this is all pretty intriguing, but I want to have a slight coda and take this back to Appledore. I've talked about temperature per se. What I haven't talked about is what is changing other than kelps. How is the system as a whole shifting and changing? I haven't talked really at all about a lot of the invasive species changes happening within the Gulf of Maine. Some of the work, real pioneering work on that, been done by Larry Harris, Jim Coyer, Phil Levin, and others on changes within the subtidal. So let's have a little coda here and think about how algal communities are changing, and in particular around Appledore. So for this, a number of years ago, Jim Coyer made me wise to the existence of some beautiful, amazing natural history journals taken by Art Borror during his time at the Shoals Marine Lab. These are now archived at UNH, and I think available to any of you who are interested in looking at them. They are just a treasure trove, just some of the most amazing pieces I've ever seen. Within them, as his students ran their intertidal transects, Art would boat around the island, looking over the side, and record what he saw on the seafloor, and built these maps. So, this is work that I'm just starting to play around with a little bit, but I'm really interested in showing you here today how the island has indeed changed over time. And so starting in 1982, we see a real mix of different algal communities. So this is the earliest map from his journals. We see in red areas of Alaria esculenta, dominating a lot of the exposed side of the island. We see Laminaria digitata, another kelp, really dominating the northern end of the island. We see Saccharina latissima, or then Laminaria saccharinea and Laminaria longicurus in some of the calmer areas of the island. But we do see the beginning of barrens. This is, again, the early 80s. And so this is when that barrens regime started to really kick up. And it is indeed kicking up off of Appledore. So we have this very kelpy area with hints of urchins to come. In 84, on a resurvey, this is, again, a little seat of the pants qualitative, but there really seem to be more urchin barrens within these maps, as well as some species shifts. So you see that Alaria esculenta, still in red, has really been reduced to one of the most exposed points on the island off of Devil's Glen. We can see some mixed reds showing up in areas they weren't previously present. There are still a variety of kelps in a number of different areas around the island, but there are more urchin barrens. There is greater area. And this trend continues into the following year. In 1987, you just have an enormous amount of urchin barrens. So those are in green here. You can eyeball this to begin with. You can see barrens in many of the areas that were previously kelpy. You still see that tiny little point of Alaria esculenta by Devil's Glen. You see a mix of reds in some places. Kelps are still present. We've never been kelp free, but man, you see a lot of urchin barren, including in some of the very exposed sites on the island. I was actually shocked at this one to see Babs Cove, which is like our big kelp bed that we see every year, was largely barren within this survey, barren and red. This is also the first time that we see Codium fragile on one of these maps, Dead Man's Finger, and you can see it off of Babs Rock. Sorry, Broad Cove is where you see it. Oh, it is late. In 2014, I was lucky enough to go out with Jim, who humored me and let me lean over the side of a Zodiac as we drove around and peeked over the side. And what you see in 2014 is something a little bit different. So this is, roughly nearly 20 years after that last survey, and things are quite different. Alaria esculenta is still over by Devil's Glen, but not really anywhere else. We see a resurgence in Saccharina latissima. You'll even see I wrote Big Kelp Bed over in Broad Cove. But you see Saccharina latissima occurring in places that formerly were either a Laminaria digitata or Alaria esculenta. So something is really different there. You also have a great deal of the island now dominated by a mix of red algae, predominantly Heterosiphonia in protected places and more, well, Heterosiphonia at the time, sorry, it's Dasisiphonia now, but various other polysiphonate and algae, as well as chondrus in other areas. You do find a number of mixed stands, and I'd say that's kind of a lot of what we see now, particularly on that northwest side of Appledore. This is what the subtidal looked like in a number of places. So we have this rise of these red turfs. Notice I mentioned there were no urchin barrens. I haven't really seen many urchin barrens in the Gulf of Maine since I started working here again in 2013. But this is a photograph of one of our sites where you can see this big fluffy Dasisiphonia turf. And in fact, this turfification is something that's happening around the world. So we've watched Appledore go from a predominantly kelp-dominated island to one that's a mix of barrens and kelps to one that is now sort of highly invaded and covered by these turfs. And as I mentioned before, turfs really are on the rise around the world. There's a beautiful review of this by Karen Philby-Dexter and Thomas Wernberg, showing that throughout the world, we're seeing warming heat wave storms, acidification, and other drivers changing a number of kelp forest ecosystems into these red turf beds. Jen Dykstra's work really beautifully elucidates some of the consequences of this, but we are really seeing some of these shifts relative to historical baselines. So, okay, let me wrap this all together. So what is the future of both the world and of Appledore? Well, one, climate change and how it interacts with other human impacts really is the story. That within these kelp forest ecosystems, as resilient as they can be, you cannot get away from climate change, and you really can't get away from how climate change can interact with other human impacts. But what comes after is still hazy. It may be fluffy, it may be red, but we really do need to know when and where the subtidal is going to change, and more importantly, what will happen to the goods and services that are provided by our current kelp forest ecosystems. Within the Gulf of Maine, we know some of that, but I'd say more is unclear. And it behooves us to understand the importance of our big, beautiful brown algae as we go forward. So I'll end just by thanking a variety of collaborators and colleagues, the Keen One Network, my lab and team dive within it, the NC's Kelp and Climate Change Working Group, as well as a variety of kelp forest collaborators on other projects. I'd like to thank my funding agencies, everyone from NC's to UMass Boston, to both MIT and Woods Hole Sea Grant and NASA. I'd also really like to thank SML. Shoals has been my home for a long time, and just coming back there every summer is an extreme delight. This summer has been painful to not be up there, and I am so looking forward to coming back next year, getting our surveys going and bringing out a new crop of students. And another theme that I hope some of you have seen running through this talk is a huge thanks to Jim. He really got me going in the subtidal and helped make me the researcher who I am. So huge thanks to Jim. It's been a humbling experience to step into your fins, and it's just been delightful to have a lifelong relationship and collaboration with you. So I'll end there, and I'll take any questions. Thanks.
That was awesome. Thank you so much. I feel like I learned a lot right there. So of course we have some questions from Jim too. So I'm excited to ask you these questions. So the first one is, how much of the worldwide variability in kelp density and distribution is due to lumping kelp species, which may have different reproductive output, recruitment, physiology, depth preferences?
Yeah, that's a great question, and definitely some of it. I'd say our analysis is a broad pass. There are some places in the world where we know that the story, for example, if you think about the UK and Ireland, right now, they're undergoing an enormous transition from a cooler water kelp species to a warmer water kelp species. So there is a huge amount of change going on there that is not necessarily reflected in the data that I presented here today. So there is a lot of variability in species in some parts of the world. In other parts of the world, not nearly as much. So for example, things that are happening in Australia and New Zealand, well, at least northern New Zealand, that's largely driven by a single species. Changes that we see happening in Tasmania, or changes that we see happening in South America, that's largely going from larger species to smaller species. So there's a huge amount of the story left to tell. In some cases, it would, if anything, flatten those curves a little bit. Yeah, this is one reason why that entire kelp time database we've made public. It's available at Temperate Reef Base. Anyone can hop in, pull it out, and not only do we have the aggregated data there, but either on Temperate Reef Base or aggregated on GitHub as a part of the project, we have the cleaned and processed data available that's down to as low a taxonomic level as we can get. And I say cleaned because 90% of that project was data cleaning. If you have made a kelp forest time series, I have looked at it. I may have judged it a little bit, but I love you all.
So there's data out there for anyone to play with. Just looking at the time, we'll take one more question here. And if anyone has questions, Jarrett said actually that he would graciously answer your question if we run out of time. So you can put it in the Q&A and we'll send it to him and he'll email you. So Jim asks, would it be useful to conduct the species richness analysis with and without introduced species?
Yeah, I think so. I think there's, again, there's a lot still to understand about the composition of the subtidal community and how it's influencing species richness. Reds are definitely playing, Dasyphonia is playing a really interesting role as Jen Dykstra's work has been showing consistently. But there are other species that I don't think we should really count out in terms of their ability to compensate for kelp loss. Again, I kind of go back to Phil's work showing that total algal biomass was really important. And I think in many of these systems, when you have, or many of these places, when you have things like Desmarestio or changes in chondrus or others, I think there's probably a lot of compensation going on there. And I think the only way we'll really be able to pull that apart is experimentally, or get those time series to be really long so we can have as many different parameters in it as possible. But I think species richness is probably going to end up being really a function of total cover, total biomass of just algae writ large, and habitat forming stuff. I think kelp here may be doing some things in a unique way, and there are some ecosystem functions that kelp is facilitating that other species don't, but I'm very excited to try and pull apart what those are and what those, more importantly, are not.
Well, we're excited for you to come take data hopefully next summer. And all you students, he's teaching underwater research, so great class. So in the interest of time, thank you, Jarrett, for answering those questions. And I think we'll have one or two to send you. Thank you for that great talk. So
my pleasure.
Next week, we will continue our Rock Talk series. Again, Tuesday, 7:30, when Steve Deneff is going to join us. He is an award-winning cinematographer, photographer, director. He focuses on the marine world. He actually lives locally and has done a lot of work and is actually right this week doing work in the shoals. And he's been all around the world. And he primarily works with scientists to help tell their story. And he's done a lot of work with National Geographic and Discovery and a lot of the obvious places. So he will bring a lot of beautiful pictures, amazing stories, and some local pictures and also global pictures. So there's more information about this entire series and other things on our website, shoalsmarinelaboratory.org. You can make a donation there if you would like to support our work. And thank you all so much for joining us on the successful No Power Loss Rock Talk. Woohoo! And take care, everyone. Thank you again, Jarrett, for a great talk. Good night, everyone.